The second of the energy-yielding nutrient classifications is lipids. Lipids are a diverse group of chemical compounds. They share one main characteristic. They do not readily dissolve in water. Lipids provide an average of 9 kilocalories per gram, which is over twice as calorically dense as carbohydrates or protein. The acceptable macronutrient distribution range for lipids is 20 to 35% of your total calories. Therefore, if you consume a 2,000 calorie per day diet, the recommended calories per day from lipids range from 400 to 700 or 44 to 78 grams. By far, the most common form of lipid that we consume is in the form of a triglyceride or a triacylglyceride. A triglyceride is formed from 3 fatty acid molecules attached to a glycerol backbone. We will examine the chemical structure of fatty acids and then consider how fatty acids combine together with glycerol to form triglycerides. A fatty acid is basically a long chain of carbons bonded together and flanked by hydrogen atoms. At one end of the molecule, the alpha end, is an acid group. At the other end, the omega end, is a methyl group. You may remember from a previous video that carbon typically forms 4 bonds. Therefore, in a long chain of carbon, 2 of the bonds are made with neighboring carbons, leaving 2 bonds for other atoms such as hydrogen. The length of these chains can range from 2 to 32 carbons long. Carbon chains less than 8 carbons are called short chain fatty acids. Chains of 8 to 12 carbons are called medium chain fatty acid, and chains of 13 to 22 carbons are called long chain. Normally, carbon chains have an even number of carbon atoms due to the way they are assembled, with 2 carbon units being added through a process called beta oxidation, but we'll cover that later. The methyl group at the end of the fatty acid is composed of a carbon with 3 hydrogen atoms. The acid group contains 1 carbon that forms a double bond with an oxygen atom and a single bond with a hydroxyl group. The electron that is shared between the oxygen and hydrogen atoms of the hydroxyl group is more so associated with the oxygen rather than the hydrogen, allowing for the hydrogen proton to dissociate from the oxygen, sometimes forming a hydrogen ion, giving it the characteristic of an organic acid. There are 3 forms of fatty acids, including saturated, monounsaturated, and polyunsaturated fatty acids. The carbons that make up the chain of a saturated fatty acid are all connected to each other by single bonds. This allows for the maximum number of hydrogens to be bound. Most fats high in saturated fatty acids, such as animal fats, remain solid at room temperature. This illustration is an 18-carbon saturated fatty acid called stearic acid. If the carbon chain of a fatty acid contains a double bond, those carbons in the chain have fewer bonds to share with hydrogen, and the chain is said to be unsaturated. A fatty acid with only 1 double bond is monounsaturated. Canola and olive oils contain a high percentage of monounsaturated fatty acids. The illustration is an 18-carbon monounsaturated fatty acid called oleic acid. A fatty acid that contains more than 1 double bond is called polyunsaturated. Corn, soybean, sunflower, and safflower oils are rich in polyunsaturated fatty acids. This is a picture of a structure of an 18-carbon polyunsaturated fatty acid with 2 double bonds called linoleic acid. Unsaturated fatty acids can exist in 2 different structural forms, the cis and the trans forms. In their natural form, monounsaturated and polyunsaturated fatty acids are usually in the cis form. By definition, the resulting cis fatty acid has the hydrogens on the same side of the carbon-carbon double bond. During certain types of food processing, which we'll discuss later in this chapter, some hydrogens are transferred to the opposite sides of the carbon-carbon double bond, creating the trans form, or a trans fatty acid. The cis bond causes the fatty acid backbone to bend, whereas the trans bond allows the backbone to remain straighter. This makes it similar to the shape of a saturated fatty acid. The Food and Nutrition Board suggests limiting intake of trans fatty acids in processed food, also referred to as trans fats, as much as possible. In addition to the cis and trans structural forms, there are also important differences regarding the location of the double bonds along the chain of carbons. If the first double bond starts with 3 carbons from the methyl or omega end of the fatty acid, it is an omega-3 fatty acid. If the first double bond is located 6 carbons from the omega ends, it is an omega-6 fatty acid. Following this scheme, an omega-9 fatty acid has its first double bond starting at the 9th carbon from the methyl end. In foods, alpha-linolenic acid is the major omega-3 fatty acid. Linoleic acid is the major omega-6 fatty acid. These are also considered essential fatty acids because we need them for a specific biological function, but we do not have the ability to make them from other fats. Oleic acid is the major omega-9 fatty acid, but it is not considered essential. Fatty acids are usually incorporated into triglycerides. A triglyceride is composed of 3 fatty acids attached to a glycerol backbone. This attachment is brought about by a condensation reaction similar to the one required to attach monosaccharides together to form disaccharides. Likewise, the fatty acids are removed from glycerol by a hydrolysis reaction that requires water. Removing one fatty acid from a triglyceride forms a diglyceride. Removing 2 fatty acids from a triglyceride forms a monoglyceride. Removing a fatty acid usually requires an enzyme, and typically those enzymes are classified as a lipase. The 3 fatty acids associated with the triglyceride do not have to be the same. They can vary by number of carbon atoms in each of the fatty acids or the degree of saturation of the fatty acids. Phospholipids are another class of lipids. Like triglycerides, they are built on a backbone of glycerol. However, at least one fatty acid is replaced with a compound containing phosphorus, and often other elements such as nitrogen. Many types of phospholipids exist in the body, especially in the brain. They form important parts of cell membranes. Lecithin is a common example of a phospholipid. The compound containing phosphorus is usually water-soluble, making phospholipids partially soluble in water. Because of this property, phospholipids are used to suspend fat droplets in water, which is called emulsification. Sterols are a class of lipids with a characteristic multi-ringed structure that makes them different from the other lipids that we already discussed. The most common example of a sterol is cholesterol. This waxy substance doesn't look like a triglyceride. It doesn't have a glycerol backbone or any fatty acids. Still, because it doesn't readily dissolve in water, it is a lipid. Among other functions, cholesterol is used to form certain hormones and bile acids and is incorporated into cell structures. The body can make all the cholesterol it needs, so cholesterol is not considered an essential nutrient. We'll talk more about the health effects of lipids in another video.